Welcome back. In today's lab, we will be covering carbohydrate fermentation, digestive exoenzymes, as well as urea hydrolysis. So let's go ahead. So what are carbohydrates? Carbohydrates are essentially sugars. What we're going to be doing today is we're going to determine what carbohydrates your unknown bacteria is capable of fermenting. Here I have 4 different types of sugars. Now you may look at them and say, well, they all look the same to me. They look very similar, right? In fact, they pretty much do look the same. However, one of them is glucose, the other one is lactose, mannitol, and sucrose. How can I tell the difference from one sugar and the other? You can't, which is why we have the caps that are color-coded, so that I can tell the difference. Looking at my little chart, my little cheat sheet that you had in your community drawer, you can tell which sugar is which. What I want you to do, what I want to show you actually, is I want you to take a close look at this carbohydrate. What do you notice? What do you notice in all of these 4 different carbohydrates, in fact? What you notice, hopefully, is, what do you think? The color, for one. Yes, the color is red. We're going to talk about why it's red. But there is something else that maybe you noticed. Do you hear that? It is a little inverted glass test tube that is inside of my larger test tube. The carbohydrates that we use for this experiment include glucose, lactose, mannitol, and sucrose. Let's discuss a couple of things that you may have noticed about the broth. For one, what you may have noticed is that our broth is red, right? The broth is red because we use the pH indicator called phenol red. So phenol red is going to detect any changes in the pH. We know that phenol red is going to be red under basic or neutral pH. Okay? I wonder what happens if there is an acidic environment or if there is an alkaline pH. Well, we're going to find out. Another thing you may have noticed is that my carbohydrate test tubes have this little glass inverted test tube. This test tube is called a Durham tube. The purpose of the Durham tube is to trap any gas that may be produced during the fermentation process. What does fermentation mean anyways? It means that the bacteria is utilizing these sugars. Okay? It's utilizing them. Sometimes when the bacteria ferments or utilizes these various sugars, it also produces gas. Sometimes it doesn't. If it does produce gas, that gas is in the form of CO2. Testing for these various carbohydrates is important because not everybody's unknown is going to basically ferment all of these carbohydrates. There are some individuals that will have an unknown that ferments maybe just glucose and mannitol. Maybe it ferments all of them. Maybe it doesn't ferment any of them at all. Everybody's unknown may have a different profile. So determining that profile will be important. Now, after we inoculate and we incubate our unknowns, incubation is usually 18 to 24 hours. This is a sensitive test, so we don't want to incubate too long. We incubate at the unknown's optimum growth temperature. And so what are the experimental results that we would expect? Let's take a look. One of the potential results would be that after inoculation and incubation, your media looks the same. What happened? Well, basically nothing. You see that your media is still at a neutral pH because your bacteria did not ferment the sugar. Therefore, these results or this result would be negative. Another potential experimental result would be as follows. Now what we see is a color change. Here is my Durham tube inside. just like it was before, right? But there has been a color change. Why has it changed to yellow? Remember that phenol red is going to be it's going to be red at a neutral pH, and it will be yellow at an acidic pH. Well, why has this media turned acidic? Some of the byproducts of fermentation, when the bacteria ferments these sugars, is an acidic environment. Therefore, we document this as A for acid or acidic, meaning that it is a positive reaction. This microorganism fermented the sugar. Another possibility for our experimental results would be as follows. This is going to be a little bit different. Yes, we still have yellow, but look what happens with the Durham tube. What I see is that my Durham tube has a sort of bubble, right? What does that due to? CO2. In this case, this microorganism fermented the sugar and it also produced carbon dioxide. We refer to these results as being A for acid and G for gas, or AG. It is a positive reaction and it also produced gas. But there is one more scenario that I want to go over with you, and that is as follows. What if, after inoculation and incubation, you get something like this? Your media turns pink. There's your Durham tube. There is no gas production in your Durham tube, but it's pink. Why is it pink? Well, one thing we know. Another thing we know about phenol red. Well, what do we know? We know that it is red at neutral pH. It is yellow at an acidic pH. And remember I asked you, I wonder what it looks like under an alkaline pH. Under an alkaline pH, it turns hot pink. Huh. Why is that? Does this mean that the bacteria fermented the sugar? No. What it does mean, however, is that the bacteria utilized other proteins, but it did not ferment the sugar. Therefore, how would we classify this experimental result? Is it positive? Is it negative? Is it A? Is it AG? What is it? It is negative. What we're looking for here is fermentation of these various carbohydrates, of the sugars, not of proteins. So we're going to be looking specifically at the production of two different exoenzymes that your unknown bacteria may or may not produce. The first test is going to be the gelatinase test. So what I have here is a gelatin test tube. Gelatin is a protein, okay, that we're going to be testing to see if your bacteria produces an enzyme that breaks down the gelatin. The other test is going to be the amylase test. This is a starch agar plate, and what we're going to try to see is if your bacteria produces the enzyme amylase that breaks down starch. So what are gelatinase and amylase? These are two different types of exoenzymes. Some bacteria are going to produce these exoenzymes. Exoenzymes are enzymes that are made inside of the cell and then secreted to its environment to break down different things. Okay? In the case of our gelatinase enzyme, the bacteria will produce gelatinase, some bacteria, which is an enzyme. Remember, they both end with ase, amylase, gelatinase. So gelatinase would be an exoenzyme that the bacteria produces inside of the cell to be secreted to the outside and break down gelatin, which is a protein. It breaks it down to its monomeric units, which would be amino acids, and that way it's easier for consumption. One thing that I want you to notice about this gelatin tube is that it is solid. See, I can put it upside down, I put it sideways, and it is solid. Okay? What do you think will happen to this gelatin if the bacteria produces an enzyme that breaks it down? Do you think it's still going to be solid? No, probably not, right? It would break it down. So the way we do this is we get a heavy inoculum, under sterile conditions, right? We get our culture, we get a heavy inoculum, flame the neck of the test tube, and we actually stab our gelatin 3/4 of the way, flame, cap it, and we incubate it at the unknown's optimum growth temperature, just like we did with the carbohydrates. Incubate them at their optimum growth temperature. But unlike the carbohydrates that require an 18 to 24-hour incubation period, the gelatin requires almost about a week and a half. It takes a while. Okay, so we're going to incubate it at 37 degrees or 25 degrees, whatever the optimum growth temperature of the unknown is, for about a week and a half. Okay, so that's the first experiment for exoenzymes. The second exoenzyme that we're going to be testing for is amylase. Amylase is an enzyme that breaks down starch. So therefore, we're using a starch agar plate. What we'll do is we're going to inoculate two different controls in a straight line, one on one side, the other one on the other side, and we're going to incubate it at 37 degrees for 48 hours. So these are our two tests for exoenzymes: the gelatin and the starch. Starch is a complex carbohydrate. If your unknown bacteria produces the enzyme amylase, it will break down the starch into simple sugars. And that way it makes it easier for being able to utilize those, right? Okay, let's go ahead and move on to the next experiment. The last experiment that we're going to talk about today is urea hydrolysis. Okay, so here is my culture for the urea hydrolysis. It also has this sort of red pigmentation to it, right? I wonder why. Why do you think it has that pigmentation? That's right. It also has a pH indicator. The pH indicator that's used for the urea hydrolysis test is phenol red. Ooh, just like our carbohydrates. So let's take a closer look at this enzyme, urease, and what it does. We start off with urea. This is the chemical formula for urea. Remember that what you're going to be doing is you're going to be inoculating a urea broth. Okay? If your bacteria produces the enzyme urease, remember that urease, basically it's a virulence factor that some bacteria utilize to establish disease, such as Helicobacter pylori. It's very good at doing that. So what does urease do? Urease is an enzyme that's going to break down this urea. Hydrolyzes it, right? Breaks down urea into what? It's going to break it down into ammonia and carbon dioxide. Ooh, well, ammonia. What happens if there is ammonia released in the media? What do we start off with? We start off with our urease tube, right? It has this pH indicator called phenol red. What if your bacteria does produce the enzyme urease? What will this test tube look like if, as a result of the bacteria producing the enzyme urease, there is ammonia that's released? What does that do to the pH? Remember that phenol red is a pH indicator. Ammonia is going to create an alkaline environment, isn't it? That's right, it will. And so what happens is, as a result, you would have this pink color. A reflection of an alkaline pH due to the production of urease, which breaks down urea and as a result releases ammonia. Those are your experimental results. That's what to expect for the urea test. And now let's go over our final experimental results for the experiments that we ran today. Beginning with the carbohydrates. Remember that we inoculated two different cultures here. We inoculated E. coli We used the 4 sugars that we used are glucose, lactose, mannitol, and sucrose. We inoculated 2 sets of carbohydrate panels with E. coli, and another one with Proteus vulgaris. Okay? Let's analyze our experimental results after 18 to 24 hours of incubation at 37 degrees, which is the optimum growth temperature of E. coli. Take a look at that. What do you notice? Well, yeah, you notice that it is yellow. What else do you notice? Look at that. Do you see that gas bubble at the top? So what would that tell you about their experimental results? That's right, AG. Remember that that's how we document a positive reaction. Acid plus gas. What about lactose? What does that look like? Acidic. It is a positive reaction. Mannitol is also acidic. Now, when you take a look at sucrose, that looks a little bit interesting, right? Looks a little orangey. Well, one thing I know is that it is not positive because you cannot have a positive reaction if there isn't any gas production. Sorry, you cannot have a negative reaction if there is gas production. You can have a positive with gas or without gas. But can you have a negative reaction with gas? No, you cannot. You cannot have a negative reaction and have gas production because gas production would tell you that the bacteria actually fermented the sugar, right? Whenever the bacteria ferments a sugar, it doesn't always produce gas, but it can. But if the bacteria does not ferment the sugar, there is no way then that it's going to also produce or that it will produce gas. Okay, that just doesn't happen. So what we see here is sort of an ambiguous result. I would say that this is negative. But when you look at your DIFCO chart in your manual, that's on page, what page is that? 78. And you scroll down that first column of E. coli, all the way to the bottom, you'll see the experimental results for sucrose. And what you'll notice is that it has a V for variable. Oh, geez, well, that kind of is confusing. Yes, it can be a little bit confusing. That means that sometimes some strains of E. coli are positive for sucrose fermentation and sometimes they're negative. Okay, so that's what V means. And some of you will have some of that as your experimental results. That can be a little frustrating, but results are results. Now, when we take a look at Proteus, this will be maybe a little bit easier perhaps. Glucose fermentation, what would you classify that as? Well, you can see the clear bubble here, right? Therefore, that would be, that's correct, AG, acid plus gas production. Now, the next one, lactose, well, this almost looks pink to me, doesn't it? So what do you think the results are for that? Remember that phenol red will turn pink under alkaline conditions. Are we looking for alkaline conditions? No. So therefore, it is a negative reaction. Mannitol fermentation, did that happen? No, it is also negative. And finally, for sucrose, our results are A for acid. Okay. Moving on to our digestive exoenzymes, our experimental results. Remember that we started off by inoculating our starch agar with E. coli and Bacillus subtilis. What do we do then? After inoculation and incubation at 37 degrees for 48 hours, we add iodine. Well, what does iodine do? If there is still starch present in my starch plate, the iodine is going to complex with the starch. Okay, well, I'm a little bit confused. Let's back up here. If my bacteria produces the enzyme amylase, what does amylase do? Amylase is an enzyme that breaks down starch. This is a starch agar plate. So if my bacteria produces amylase, it's going to break down the starch that surrounds my bacterial colony. So how do I detect if my bacteria produce this enzyme amylase, therefore breaking down the starch that surrounds the colonies? If it breaks down the starch, then if I add my iodine, which is what the next step would be, I would flood my plate with iodine. The iodine, remember, is only going to bind to areas where starch is present, thereby giving me a sort of purplish dark color. Whoa, this looks almost black here, but if you look at the outline at the bottom, it looks purple, right? Okay. So notice that where my E. coli streak was, I can't even see the E. coli because it's flooded with the iodine. Therefore, it tells me that there was starch surrounding the E. coli because E. coli does not produce the enzyme amylase. But when you look at the Bacillus subtilis, you see that there is a sort of halo that surrounds the bacterial colony, surrounding the Bacillus subtilis. What this tells me is that Bacillus subtilis produces the enzyme amylase, and therefore this enzyme hydrolyzed or broke down or digested the starch that surrounds the bacteria. And since there isn't any starch present around the bacterial colony, the iodine does not bind there because there isn't starch. Therefore, we say that E. coli is negative for amylase. It doesn't produce the enzyme amylase. And Bacillus subtilis is positive for amylase production. Okay? The second exoenzyme that we tested today was gelatinase. This is the gelatinase test. Now, I showed you my gelatin media. It was solid. We inoculated it with the heavy inoculum of our unknown, or my controls in this case. Remember that in your manual, you're going to use the controls that I list here on this experiment. What do you think is going to happen to the gelatin if it's been incubating at 37 degrees for a week and a half? 37 degrees is very warm, right? What's going to happen to the gelatin? It's going to melt. It's going to be liquid. What happens if my bacteria produces the enzyme gelatinase? It breaks down the gelatin, making it liquid. So how will I know if my gelatinase test tube is liquid because the bacteria produced the enzyme, or if it's liquid because it's been at 37 degrees? I wouldn't know. Therefore, the first thing I do is, after removing them from the incubation at 37 degrees, I put them in an ice bucket for about 20 minutes. If after 20 minutes my media remains liquid, then I can say that it is positive for gelatinase. However, if after 20 minutes my media solidifies, that means that it is negative for gelatinase. In today's experiment, the unknowns, or the controls that I use rather, are Staphylococcus aureus and Bacillus subtilis. Here on the left, what you notice is that Staphylococcus aureus, after 20 minutes of putting it on an ice bath, my gelatin solidifies. Well, what do you think that means? If the gelatin solidifies, that tells me that Staphylococcus aureus is negative for gelatinase. The bacteria does not produce the enzyme gelatinase, and that's therefore the gelatin was able to solidify upon freezing or upon putting it on the ice. Bacillus subtilis, however, after putting it on ice for 20 minutes, notice that when I put it vertical or horizontal here, you see that it's liquid now, right? It remained liquid even after putting it on an ice. Therefore, these experimental results are positive. Yeah? Okay. The final experiment that we're going to talk about today is the urea hydrolysis. We started off by inoculating a urea broth with E. coli and Proteus vulgaris. After 48 hours of incubation at 37 degrees, we see our experimental results. Remember that urea hydrolysis test, the urea broth has a pH indicator called, what was it called again? Phenol red. Under acidic conditions, what does phenol red look like? Yellow. Under alkaline conditions, what does our phenol red look like? So what will a positive reaction or a negative reaction look like in the urea hydrolysis test? Well, we got to think back about what this enzyme urease does. Urease is an enzyme that's going to break down urea, and in the process, what does it do? It releases ammonia. Here we see E. coli. E. coli is going to remain yellow, or it changes to yellow. Sometimes even if it doesn't change color, we say that this is a negative reaction. But when we look at Proteus, we see that Proteus is actually hot pink. It's a beautiful color. Why has it changed to pink? Well, you got to remember, what kind of environment will ammonia create? It will create an alkaline environment. Therefore, Proteus is positive for urea hydrolysis. Okay? So we sort of have a different result. A positive reaction for urea is going to be much different. It's going to look much different than for your carbohydrate fermentation. And this concludes today's lab.